Hello, everyone, and welcome to lecture 6 for this chapter. Let's get into it. In this lecture, we're going to talk about the neural control of emotion. So there's 3 forms of emotional experience that suggest the influence of different neural bases of emotions. There's the autonomic response, such as an increased heart rate, where the hypothalamus and associated structures, as well as the ENS, are involved. There's also the subjective feelings, such as the feeling of fear, where the neuroanatomical structures related to that are the amygdala and parts of the frontal lobe. Then there are cognitions, which are thoughts about the experiences, and that is involved with the cerebral cortex broadly. Now, some of the challenges that take place when we're thinking about the neural mechanisms related to emotions is that emotion words are not the emotion. It's a hard concept to grasp, but it's essentially there's this phenomenological experience of what an emotion is, what the feeling is, and that is different from some of the words that are used. So emotion occurs in contexts that strongly encourage brain activity, which is why often memories can be more pertinent after these highly emotional experiences, whether good or bad or whatever else. And fMRI measures do not afford normal contexts for having emotional experiences. So let's go ahead and talk about this a bit more because I know there's a lot that was in there. Let's start with some of the theories of emotion to begin. There is the constructivist theory, or the James Lange theory. This suggests that the brain interprets physiological changes as an emotion and produces a cognitive response to autonomic information. That response then varies with the context in which the autonomic arousal occurs, including the effect on the gut via the ENS. The brain concocts a story to explain the bodily reactions. A modern view of constructed emotion, is that the primary role of the brain in emotional behavior is the integration of all the body's resources to meet the organism's need for action, such as the growth, survival, and reproduction. Brain structures receive input from the periphery to generate then effective feelings. Now, the constructivist researchers propose then that the brain structures, including the anterior cingulate cortex, the ventromedial PFC, the insula, the amygdala, the ventral striatum, they receive inputs from the periphery. So those are going to be things like the skin, the ANS, and other body organs to then generate some of these effective emotional feelings, essentially. There's also the appraisal theory. This defines emotions as processes rather than states, and it views emotional episodes as the activity of several biological subsystems or components. There's the appraisal component, in which there is an evaluation of the context. There are the somatic components or the physiologic effects. There's the behavioral component, and there's the feeling or feeling. Then there are these appraisals, which leads to the process of differentiation, which encompasses action tendencies, physiological responses, and motor expression. Events interact in the brain to induce nonverbal feelings that are then categorized and labeled with emotional terms, such as good or bad feelings. These components then form the representation of the emotional event. So emotions are elicited either by external events or objects or by internal processes such as imagination or your memory. You might remember something and it'll lead to some emotions rising within you. There's then this appraisal process that leads to the process of differentiation, where then there's these action tendencies and these physiological responses, motor expressions, and then these events are theorized to interact in the brain to then induce the phenomenal experience of the emotional event. There are also neuropsychological theories, such as the right hemisphere plays a major role in producing strong emotions, especially emotions regarded as negative, whereas the left hemisphere generates emotional feelings, and the left hemisphere then interprets those feelings. Oh, there is a danger in overemphasizing laterality at the expense of diminishing the bilateral roles of regions such as the prefrontal cortex, anterior cingulate cortex, and amygdala. In many ways, the role of the cerebral site, the PFC, is far greater than that of the cerebral side. And I think that overall, this comes into play when people talk about, oh, I'm right-brained, I'm left-brained, and they like to think about it in that way. It's important to note you are most likely both. And you're more a combination of these two than one or the other, as far as some of these things go. So I think that that is just important to think about, that it does seem as though there is a little bit of a tendency in the organization of hemispheres with some of these emotional experiences, but by and large, we're experiencing a combination of those every single day. And I don't think that it's probably the best for us to think of ourselves as one or the other. When we're talking about that emotion in the limbic circuit, so the limbic lobe was first described by Paul Broca. The term now that's used is called the limbic system. It's formed from a 3 and 4 layered cortex, which lies adjacent to the 6 layered neocortex. The allocortex, that limbic system cortex, encompasses the cingulate gyrus and the hippocampal formation. So the hippocampal formation includes the hippocampus. You may have heard of that before. It's a pretty popular brain structure. That includes all the cortical structures that are important in species-typical behaviors, memory, and spatial navigation. It's also vulnerable to the effects of stress. And there's the parahippocampal cortex that is included in the hippocampal formation as well. So here's a figure that's showing Broca's concept of the limbic system. We can see that encircling the brainstem, the limbic system, as described by Broca, consists of the cingulate gyrus, the hippocampal formation, which is, again, recap, the hippocampus and the parahippocampal cortex. And we see these here, right? There's also then the amygdala, which is right at the end of the hippocampal formation, the mammillothalamic tract, which we can see here, and the anterior thalamus. Most limbic system structures, especially the amygdala and the hypothalamus, participate in emotional behaviors. They perform important roles in various motivated behaviors, especially in motivating species-typical behaviors such as feeding and sexual activity. Now, in figure A, up over here, this is the contemporary conception of the limbic system. It's an interconnected network of structures. There's this circuit that's been called the Papet, pardon me, the Papet circuit, which controls emotional expression. Figure B is coded to the brain areas that are shown in part A by color. So this is a schematic represented in charts of the limbic system's major connections. And in the figure C, this is a reminder that parts A and B can be conceptualized as part of this funnel system. And then there are the outputs that through the hypothalamus then produce the emotional motivated behavior. The amygdala is the almond-shaped collection of nuclei in the limbic system, and it consists of 3 principal subdivisions. There's the cortical medial area, the basolateral area, the central area, and it receives input from all the sensory systems. The amygdala sends projections primarily to the hypothalamus and the brainstem, where it influences neural activity associated with emotions and species-typical behavior. It can also induce eating and drinking. Now, when there's the need to remove the amygdala, it reduces this syndrome called the Kluver-Bucy syndrome. And there's been some research that was done in studies with monkeys that found that in this case, there is this tameness or a loss of fear entirely there is no fear mechanism that takes place. There's this indiscriminate dietary behavior. There's significant increase in autoerotic gay and heterosexual activity with inappropriate object choice. There's a tendency to attend to and react to every visual stimulus. There's a tendency to examine all objects by using the mouth. And there's this visual agnosia, the inability to recognize objects. Now, it's important that although the Kluver-Bucy syndrome is not common in humans, bilateral temporal lobectomies are rare, but its symptoms can be seen in people with certain forms of encephalitis or brain infection. In some cases, encephalitis centered on the base of the brain can damage both temporal lobes, and many or can produce many of the Kluver-Bucy symptoms, especially some of the indiscriminate sexual behavior and tendency to examine objects by the mouth. But again, it's not commonly found in humans. And a lot of these symptoms were shown by these prescribed surgeries on monkeys to examine the behaviors that take place when these brain areas are removed. Now, it's important to recognize that the survival of species really requires a functioning amygdala. So electrical stimulation produces an autonomic response, such as increased blood pressure and arousal and a feeling of fear. The olfactory information then connects directly to the amygdala in the human brain. Now, we need some of this fear in our lives in order to continue to exist. If we don't have any fear, then as a child, you may run into the street without looking both ways because you're not afraid of the consequences of what might happen if you don't, or you might end up trying to go skydiving without an instructor because you think that, oh, it won't be a big deal, it's not that scary. And there's also an example of, if you've seen the documentary Free Solo, that's somebody who he has a functioning amygdala, but especially in the context of the climbing without any ropes to help catch him in case he falls, he's not getting this autonomic response or the feeling of fear in that context. When, at least if you put me on the Half Dome in Yosemite, I would be clinging to that wall in a lot of fear. The amygdala also influences autonomic and hormonal responses via the hypothalamus, and it influences conscious awareness of positive and negative consequences via the prefrontal cortex. The prefrontal cortex contributes to specifying the goals of movement. It controls the selection of movements appropriate to the particular time and context. The selection may be cued by internal information, such as memory and emotion, or it may be a response to the context and environmental information. The orbital frontal area has direct connections with the amygdala and hypothalamus. It's responsible for the conscious awareness of emotional states produced by the rest of the limbic system, especially the amygdala. The prefrontal cortex is a large area of the brain. It's made up of several functionally distinct regions. Some of the general areas include the dorsolateral region, the orbital frontal cortex, and the ventral medial PFC. Pre-FC meaning the prefrontal cortex, of course. So the prefrontal cortex receives input via connections from the amygdala, dorsal medial thalamus, sensory association cortex, posterior parietal cortex, and dopaminergic cells of the ventral tegmental area. Dopaminergic input regulates how prefrontal neurons react to stimuli, including emotional ones. And abnormalities in dopaminergic projections may account for some disorders, including schizophrenia. The prefrontal cortex sends connections to the amygdala and hypothalamus. It influences the ANS and the ENS. And it also sends connections to the sensory association cortex, the posterior parietal cortex, the cingulate cortex, basal ganglia, and premotor cortex. And it can have an influence on cognitive behavior. This image is really just showing you a breakdown of the gross subdivisions of the frontal lobe and the prefrontal cortex, just so you again have an idea of where some of those areas are. As a reminder too, whenever we're talking about neuroanatomy, one of the benefits is that the terms that are used are usually pretty pretty descriptive of where in the brain these areas actually are. So if we're looking here, dorsal, medial, prefrontal cortex. So prefrontal cortex over here, dorsal medial in the middle. This is a medial view, dorsal on the top. The anterior cingulate cortex, anterior area, and the ventromedial prefrontal cortex. So again, still prefrontal, medial, ventral. In this figure, we are able to see some of the prefrontal connections. So the prefrontal cortex, it receives inputs, which are the red arrows that are shown in this illustration, and it sends outputs, which are going to be those blue arrows in the illustration, to areas on the diagram to the right. So like the amygdala, the frontal lobes, they receive highly processed information from all sensory areas, and many neurons in the prefrontal cortex, like those in the amygdala, are multimodal. The prefrontal cortex receives input via connections from the amygdala, dorsal medial thalamus, sensory association cortex, posterior parietal cortex, and dopaminergic cells of the ventral tegmental area. Dopaminergic input is important for regulating how prefrontal neurons react to stimuli, including emotional ones. And abnormalities in this dopaminergic projection may account for some disorders. Again, we talked about schizophrenia being one, in which people show little emotional reaction to typically arousing stimuli. In summary, the role of the frontal lobes in selecting behaviors is important for considering what causes behavior. The frontal lobes act a lot like a composer, but instead of selecting the notes and the instruments, instead it's selecting our actions. Not surprisingly, the frontal lobes are sometimes described as housing the brain's executive functions. And then here, just to kind of put an end to this lecture, there was this researcher, Carlisle Jacobson, who was investigating the effects of frontal lobotomy on the cognitive capacities of 2 chimpanzees. And this person found that damage to the prefrontal cortex has severe effects on social and emotional behavior, as you might expect, I suppose. And Edgus Moniz performed frontal lobotomies. This is the first psychosurgery to alter behavior, and it was later refined by Walter Freeman. So, yeah, we can see in this image here, this is an image of transorbital leucotomy. And so in this procedure, leucotomy is inserted through the bone of the eye socket to disconnect the orbital frontal cortex from the rest of the brain that takes place there, which is interesting. All right, that is the end of part 6 for this chapter, and we will move right along into part 7. So let's go ahead and get into lecture 7. This is going to be a short lecture just about reward. So rewards are not a single thing, but they rather involve several different components. The reward mechanism may have evolved to increase the adaptive fitness of the entire species and individual members of a species. Kent Burridge and Terry Robinson, they posit that reward has 3 main components. There's the learning component. It's all about the rewards and the cues that predict their availability. There's that motivation for these rewards and the cues associated with them, wanting the reward. Then there is affection, which are the hedonic responses to the actual pleasure of the reward. So it's the likingness of the reward. So there's a challenge for neuroscience in understanding how brain mechanisms generate each of these components. We want to consider why we increase contact with stimulus like chocolate, for example. And there's 3 independent factors that are our work. Our knowledge of where the chocolate might be, our desire to eat the chocolate, like that wantingness, the motivation, right? And the pleasurable effect of actually eating the chocolate, liking the chocolate. And trying to understand where do these events end up actually occurring in the brain. James Olds and Peter Milner, they were looking at intracranial self-stimulation or brain stimulation reward. So in their study, they had rats press a bar to self-administer electrical stimulation to specific sites in the brain. The brain stimulation activates the system's underlying reward. So stimulation along the MFB tract activates fibers that form the ascending pathways from dopamine-producing cells of the midbrain tegmentum. This mesolimbic dopamine pathway sends terminals to sites that include the nucleus accumbens in the basal ganglia and the prefrontal cortex. Reward itself has two independent processes. There's the wanting, incentive, and the liking, or the evaluation of pleasure. And wanting and usually liking, usually but not always, occur together, which is important. Then Robinson and Berridge, they had shown that wanting and liking are actually these separable neural systems. So wanting, that involves dopamine, whereas the liking involves opioid and benzodiazepine GABA systems. In this figure here, this is helping illustrate. So the first clear indication that dopamine is not the pleasure molecule came from studies by Berridge and Robinson all the way back in 1998. And in this figure, we see that there are these axons that are emanating from the ventral tegmentum in the blue arrows that project diffusely through the brain. Dopamine release in these mesolimbic pathways has a role in the feelings of reward and pleasure. The nucleus accumbens is critical structure in this reward system. The mesolimbic dopamine pathway sends terminals to sites that include the nucleus accumbens in the basal ganglia and the prefrontal cortex, blocking dopamine with drugs such as neuroleptics appear to take the pleasure out of typically rewarding events such as self-stimulation and amphetamine. And Roy Wise proposed that the dopamine synapses represent a place where sensory inputs are translated into hedonic messages that we end up experiencing as pleasure. Thus, we're born with this well-known idea that dopamine is the pleasure transmitter, but unfortunately, this idea is actually believed to be incorrect. Now, when we have a rat with a self-stimulation electrode in the lateral hypothalamus, it will often eat heartily while the stimulation is on. So during stimulation, rats react more aversively to tastes such as sugar and salt than when the stimulation is off. So the stimulation increased the wanting, but not the liking. Thus, the reward system processes for wanting and liking are independent of one another. This figure helps to show that a bit more clearly as well too. So wanting is mediated by the extensive brain system, including dopamine projections, which is the lighter shaded blue arrows. In contrast, liking is mediated by a more restricted brain system in these small hedonic hotspots, which are shown in red. And finally, it's notable that pleasure is not merely a sensation or thought, but it's best conceived as an additional hedonic gloss, the gloss that creates the pleasure versus displeasure affect. The hedonic gloss of objects or events is generated in the brain by activity in networks of hedonic hotspots for pleasure or cold spots for displeasure. In the past decade, deep brain stimulation has developed as a therapeutic technique for a wide range of conditions, including chronic pain, depression, and Parkinson's disease. And I think that's, again, just interesting to think about how some of this could be used and applied in clinical settings as well. So I hope you found that interesting too. That is the end of part 7 and the end of the lectures for this week. Have a great rest of your week. Bye.